Okay, so the last time we were talking, we were presenting an experiment that looked at what's the role of APC in the mitotic transition, and we used the destruction box peptide that was going to work as a competitive inhibitor to inhibit APC. And what we discovered is that if you inhibit APC with enough of this competitive inhibitor, so you shut it down, and APC is that ubiquitin ligase that's degrading cyclin B, that in fact you arrest at an earlier stage of mitosis, which is the metaphase to anaphase transition. And so remember, the metaphase is where the chromosomes are lined up, and you've got replicated chromosomes. So this is like chromosome. Here we're dealing with chromosome 1 from the let's say from the father, and here's one copy, and here's a copy that's made in S phase. Okay, and they're going to be segregated from each other. They're all being lined up. Okay, so we're talking about the chromosomes are not separating to go from metaphase to anaphase. Okay. All right, so now we're going to go over that mechanism. What is the substrate that has to be degraded at the metaphase to anaphase transition to allow those chromosomes to separate? Okay, so at this point, what's happening is you got to keep in mind that these chromosomes, they're not showing you here, so these chromosomes are under tension. They're being pulled apart because they have kinetochores that are attached to microtubules. The microtubules are pulling them in the opposite direction. Okay, but they're not going anywhere because the chromosomes are connected to each other through proteins called cohesins. So these are similar in structure to the condensins, which we went over before. So the condensins, we're gonna somehow loop together DNA and hold it in position to allow the chromosomes to be condensed. Cohesins have a similar structure, except now what they're doing is they're going to trap DNA, but they're trapping DNA presumably from both chromosomes. Okay, so if you put in the one chromosome DNA and the other chromosome DNA, well, clearly they're linked together now. So there's going to be, many of these cohesins holding together the chromosomes. And so even though they're being pulled in opposite direction by microtubules, they're not going anywhere because they're zippered up. All right. So what APC is doing is there's a protein called separase, which is going to separate the chromosomes by cutting the cohesin. Okay, and it's going to actually cut. So these cohesins have a cap, and for the mitotic cohesin, the cap is SSC1 and SCC3. It's going to cut that. Okay, it's going to cut SCC1. It's going to cut it. It's not degrading the whole thing. It's just cutting it. Okay, that will allow any DNA that's trapped in there to come out. All right, so it's going to cut the cohesins, and then once these chromosomes are not connected to each other, now the microtubules can pull them in opposite directions. Okay, but APC is not targeting separase for degradation. It's targeting an inhibitor of separase called securin. So, you can think of it like securin securing the separase to make sure it's not active. And so securin is going to be degraded, and that's going to release separase, which is a protease, will then cut the cohesin on that cap, allowing the DNA strands to be released, and because they're being pulled by the microtubules, now you'll get anaphase. Okay, so that's the critical substrate at this transition. Okay, and this is just some kind of logic of what's going on. So securin, which is what's holding the separase and inhibiting it, has a destruction box. Okay, so basically what happens is APC is going to get activated, and everything with the destruction box, that's its degron, is going to be degraded. Degron just means what it recognizes on the substrate. Okay, so if you express securin that has its destruction box removed, well, the destruction box is how APC is targeting it for degradation. And so in this situation, it can't be degraded by APC. So you basically stabilize securin if you remove its destruction box. So that's gonna lead to a metaphase arrest. And that makes sense, right? So basically, if securin can't be degraded by APC because it doesn't have a destruction box, it'll just hang on to separase. Separase will never get active, and you're not going to separate the chromosomes. So you're going to rest at metaphase with the chromosomes together. Okay, if you don't have APC mutants, you're not going to have any sister chromatid separation. That makes sense too, right? We already showed that if you inhibit APC, you get stuck at metaphase. And sister chromatid, what is that, right? Sister chromatid just means you have chromosome 1 from the father, chromosome 1 from the mother, chromosome 3 from the father, chromosome 3 from the mother, like that, right? You get one pair of chromosomes. During S phase, they separate, they replicate. So now you have 2 chromosomes. That's what's being separated during mitosis. Those are called sister chromatids. Okay, sister chromatids are the replicated chromosomes that are going to be separated from each other and that line up, during mitosis. Okay, so APC mutants, no sister chromatid separation. Another way of saying you're stuck in metaphase. You're not going to go to anaphase. But if you inactivate APC in yeast and you have a securin mutant that is not, so securin's not present, it doesn't make any difference. Then you're going to get separation. Okay, so what's happening here is what that's saying is that you need APC to go from metaphase to anaphase. But you don't need APC to go from metaphase to anaphase if securin's not present. If this separase will be released by itself. I mean, if it just on its own can be activated. Okay. So therefore, that securin is the critical substrate at this transition for APC. Okay. Now, I mean, if you ever study this, you'll see that that's actually not true in mammalian cells. But, we're not going to get into that for this class. For mammalian cells, actually degrading cyclin B is actually important for this transition too, and I could explain it later. But that's not, for this class, we're going to keep things simple. This is what everybody thought for like decades, so we'll just go with that. Okay, so if you have securin mutants, so now the separase is around all the time, you're actually going to have normal timing of chromatid separation. It's not going to occur sooner. Okay, so even if a securin is not around anyway, you're going to have this thing. This thing is only going to get activated at the right time. And it turns out that, in fact, in mammalian cells, cyclin B has to be degraded to allow separase to become active because it's also being phosphorylated by the CDK1 cyclin. Mm-hmm. Mm-hmm.
You said that that separates sister chromatids, and you said that it cuts the mitotic cohesion.
Mm-hmm.
Doesn't it have to, like sister chromatid separation only occurs in like metaphase, anaphase 2, right?
So metaphase to anaphase transition. So we're not talking about are you talking about meiosis? We're not talking about meiosis. I mean, so basically in meiosis you have two divisions. You have metaphase to anaphase 1 and metaphase to anaphase 2, but we're not talking about meiosis. We're just talking about mitosis. So this is more information than you need, but during meiosis they're using another cohesin that has a different. So it has the same backbone, the same alpha helices, but it uses a different cap. So in meiosis, then separase is also involved, but it's cleaving Rec8, so it's a different one. It's a different. It's just more information that we're not covering in the class. Okay. All right. So anyway, so the other mechanisms are, in fact, cyclin B is also has to be degraded so that the separase can become active. Okay. So now we're going to talk about, if you remember from that movie, that chromosome movie where we were looking at Nemarsky microscopy, and we had to wait for that last chromosome to come in, and the cell didn't go through metaphase to anaphase until that last chromosome came in, and then as soon as it hit that plane, then it went. Okay, so we're going to talk about that, and that's going to be a checkpoint that's going to, it has different names, but one is mitotic spindle checkpoint, okay? And it's going to wait until everybody is lined up at the metaphase plate. So it's working right here. We had already talked about this. Well, we're going to talk about this later, this unreplicated DNA checkpoint. But now we're going to talk about this one, which is this metaphase to anaphase checkpoint, to make sure that everybody is lined up. Okay. All right. So this is a hand drawing, and it's just going to be going over the different things that have to happen for this checkpoint to be satisfied. Okay, so the kinetochore is where microtubules are attached. So you can have, if you have a chromosome that is unattached on one of the kinetochores, clearly it's not going to get lined up at the metaphase plate, okay? So it has several things that's happening. So one is it doesn't have an phosphorylation at, it has phosphorylation at the unattached kinetochore. The attached one doesn't have phosphorylation, but the unattached one has it. And the way that they determine this is somebody just took mitotic cells and injected it into rabbits to make antibodies. So the antibody's saying, oh, this is foreign, I'm going to make an antibody against it. And then they realized that one of the antibodies was only recognizing things that were phosphorylated. They could tell that because when they put that antibody on, it only lit up mitotic cells, but if they treated with phosphatase to get rid of phosphates, then it disappeared. It wouldn't recognize it anymore. Okay? So that's called a phosphoepitope. Epitope is what the antibody binds. Phosphoepitope means that it's phosphorylation. It's recognizing a phosphorylated protein. And this is just the name of the antibody, 3F3/2. Okay, so that antibody is recognizing something phosphorylated, but only on the unattached kinetochore. So that means that what we're talking about is what goes along with that, that checkpoint. Okay, and then it's got these checkpoint proteins, MAD2 and BUB3, are localized to the unattached kinetochore. So as we'll see, they're actually involved in causing this checkpoint. Okay, so they're there. So then you're saying, well, what will get these things off of it? Okay, so it turns out that if you add tension with a microneedle, then that phosphoepitope, which was there, okay, disappears. So that phosphoepitope is actually paying attention to whether or not there's tension. But MAD2 and BUB3 are still there, so that they're still saying, yeah, there's something not right about the situation, and we're going to stop it. And then, if you say, okay, well, what if what they're looking for, MAD2 and BUB3, is whether or not there's microtubules involved. So then you add the drug Taxol, and so this stabilizes microtubules, but because there's no, dynamics to the microtubules anymore, they can't, be unprocessed. They can't be taken apart. Okay, there's no tension. So basically, chromosomes are hit with microtubules, but there's no dynamic tension. There's no movement of the microtubules. So now you've got microtubules, but there's no tension. Okay, they're not being pulled. They're just being attached to microtubules. So in this case, the phosphoepitope is there because it's paying attention to whether or not there's tension or not. But MAD2 and BUB3 disappear. Okay? So they're evidently paying attention to whether or not there's microtubules. So there's actually two signals that you need to get to satisfy this checkpoint. So one is you have to have microtubules attached. Okay? And if microtubules are attached, MAD2 and BUB3 disappear. And then you have to have them under tension, and so then that phosphoepitope disappears. Okay? So you can understand why you have to attach microtubules, because unless you attach microtubules, then you're not going to separate. There's no way you can line it up, right? Metaphase plate. But why would the cell evolve to require tension? And unfortunately, my document camera. Oh, it's working now. How did they do that? Really? That's cool. See, you're very efficient.
Thank you.
They didn't even have to come here.
I didn't have a meeting.
All right. So they actually. I'll just give you some background how they did this, because clearly, if you're looking at this, you're like, there's no way in hell. Okay, this is a glass needle. Okay, they're taking a glass needle, and you're like, there's no way in hell that they did that, right? I mean, what is it, a worm? There's no way that they did that and pulled on it, And you're right. That's sort of ridiculous. So actually they use grasshopper. And you're like, grasshopper? Who would study grasshopper? why would you do experiments with grasshopper? Okay, but like I told you before, there's almost always a reason that people choose these different weird organisms for the thing. And the reason they chose grasshopper is because grasshopper has circular chromosomes. So while they do have this kinetochore here, right, where microtubules are going to be attached, the whole thing is a big circle. So now they come in here, and then they use a needle. They can pull on that. Okay, so that's why they use grasshopper. So in fact, they didn't just go into the chromosome and try to pull it. They actually use grasshopper, and that's how they got the tension. Okay, that was just an aside. It has nothing to do with this class. You were probably wondering about it or not, depending on whether you're paying attention. But now here's the question for you. So the question is, why do they need to have tension? Why would the cell have evolved to require tension along with microtubules to satisfy the checkpoint? Like, what would happen would there be a situation where you don't have tension, but you've got microtubules attached? Yeah. Oh, you got it. Were you raising your hand? No. Okay. Are you talking about like this? Yeah, so this is an unattached kinetochore. So basically, all chromosomes are not going to be attached to microtubules initially. And what happened here is maybe this is the last chromosome or whatever, and microtubule is attached on this side, but hasn't attached on this side yet. And, until you have that situation where they're both attached with tension, they're not going to go through the metaphase to anaphase transition. So, just in general, because, there's no way that you're going to get attachment at the exact microsecond for both of them. You're always going to have a situation where, it attaches on one side first, then the other side. Okay? And we actually saw, we saw an example of that chromosome that got attached way late and came in way late in that movie that I showed. So why would you need to have tension? Yeah. No, as soon as they're attached on both sides, they're under tension. Yeah. Yeah. You mean the kinetochore? No, not really, because I think all of these microtubules are attached on the centrosome, because I think that's how they grow out. So they all grow out from the centrosome. I don't think there's any non-centrosome microtubules at that stage of mitosis that I'm aware of. But that's a good idea. If that were the case, that would definitely be a reason to do it. But I don't think that's actually something that actually happens. It's actually a very hard question. Only super smart people can get it. Which one of you are super smart? All right, do you give up? Do you want me to tell you? Okay, so the question is, you've got a situation where, okay, so evolutionarily, you have to have microtubules attached, but you've also got to have it under tension. So what's the reason that you need to have it under tension in addition to having microtubules attached? What would be a scenario where you might have microtubules attached, but there's no tension? Because clearly that's the situation they're trying to avoid by having the tension, right? Otherwise, you would never have, let me put it this way. If it's redundant, evolutionarily, you would only need one or the other of these mechanisms. You wouldn't need both. But there's a situation that you have the kinetochores, which is where they attach, and you've got microtubules on both of them, but there's no tension. So let me go backwards. So what's the whole purpose of this checkpoint? What's the purpose of why have the mitotic spindle assembly checkpoint? What would bad happen if you didn't have the checkpoint? This is a more basic question. You said non-disjunction? Yeah, so that's right. So you'd have non-disjunction. And what does non-disjunction mean? So it means that okay, what it means is that, like, under normal situation, you're going to end up with, these things going to each side like this. Okay, so you've got one copy of each. But if you have a non-disjunction situation, what you end up with is okay, non-disjunction means that these didn't disjoin from each other. And now, let's say this is chromosome 3 from the father. Well, this cell got both of the copies, and this cell didn't get any. Okay, so that's what you're trying to avoid. You're trying to avoid this situation. That's why you have to have them all lined up with tension before you do it. So what would be a situation where you would get non-disjunction, where you have microtubules, but there's no tension? Yeah.
Do you have microtubules from the same side?
Yeah, that's exactly correct. So the answer is microtubules from the same side. So now there's no tension because, there's tension because they're being pulled in opposite directions. But now they're being pulled in the same way. They're just moving. There's no tension, okay? So that's exactly what you're trying to avoid. And this is called a meroteelic attachment. That just means that both kinetochores are attached to the same side. So until these kind of things are resolved, you can't go to the next position, okay? You can't get to the metaphase, anaphase. You can't actually separate the chromosomes. So that's absolutely correct. Okay, so and then you're saying, well, how does this whole tension thing work? Okay, so this is a slide you don't have. I'm going to post on an updated -2, and I already did that for one previous lecture, I think cell cycle 1. So you probably want to download that also at some point. Okay, so it turns out that there's a kinase called Aurora B, and it's that one that's phosphorylating things at the kinetochore when there's no tension. So what's happening? Okay, so this Aurora B is in something called the chromosome passenger complex. Well, okay, that's sort of a long name. Maybe I don't need to memorize that, right? But the point is that it's located between the kinetochores, okay? And so when there's no tension, it's actually this CPC, this red thing, can actually touch the kinetochores and phosphorylate things at the kinetochore. But when there's tension, it's actually pulling the kinetochore away from the CPC structure that has the Aurora B kinase. So the kinase can no longer touch its substrates, okay? So anyway, this is what people think is happening based on, these kind of observations where they see this kind of like separation, while they're under tension. So that's the idea for how that tension thing is worked out. All right. So this is what's actually happening in this this thing in this checkpoint, and you're making something called the mitotic checkpoint complex, MCC. All right. So this thing is going to form at unattached kinetochores, and so what it does is it's going to inhibit APC. So unattached kinetochores are like an assembly point for making this structure. Okay? Now this structure is going to bind to APC and inhibit it. And as long as APC is inhibited, then nothing's going to happen. You're not going to degrade securin. You're not going to degrade cyclin B. So here we're talking about degrading securin, which releases separase. Here we're talking about cyclin B, which allows mitotic exit. So these things are not happening as long as this thing is being made by an unattached kinetochore and binds to the APC. Okay? So this is a structure of APC. So APC, I told you, is huge. It's got like 8 to 12 proteins in it. And it actually has one of the subunits recognizing substrates. and that subunit is CDC20, but it's sort of weird. So basically, CDC20 recognizes the instruction box, the part that's on substrates, and it'll bind those substrates, and then the substrates get degraded when they bind this complex. Okay, so it's binding substrates to the CDC20, and then this complex adds ubiquitin to it, and then the substrate gets degraded. What's sort of weird is that this MCC complex also contains a CDC20. So it's a different CDC20. So there's a CDC20 here, but there's another CDC20 in this complex. I don't know why evolution did that, but it did. And in fact, the two CDC20s interact with each other. So here you have CDC20, and what you see is that there's a CDC20, but now it's being bound by this MCC complex. Okay, like here they've color-coded it. So there's the CDC20 in purple that's bound to APC, but then this is the CDC20 part of this complex, and it binds it. So then you could ask yourself, well, what happens when this one finally gets attached and then lined up? How is it going to work? All the APC are inhibited, right? They're all inhibited. So how's it going to work, right? Okay, so it turns out that this thing, when it's bound here, turns out that, CDC20 normally binds substrates, and then the substrates get ubiquitinated. Well, it turns out that APC sort of thinks of this thing as a substrate, and it gets bound here, and then gets ubiquitinated, and then degraded. And then another one comes in and inhibit it, and it gets ubiquitinated and degraded, okay? So as soon as you stop making this MCC complex, APC is going to ubiquitinate the one that's on it and degrade it, okay? It's constantly ubiquitinating and degrading it, and a new one comes on to bind it, okay? So that's how it gets rid of it. That's why once you satisfy this one, once it gets attached, you stop making this complex. Well, the one that was there just gets ubiquitinated and degraded, okay? So it's relatively quickly that it'll overcome that inhibition. All right, so now we're going to talk about cytokinesis and why cytokinesis occurs after the metaphase to anaphase transition. So in mammalian cells, the cytokinesis is going to be an actin contraction. So there's actin filaments that are at the site of cytokinesis. You can see them here when you stain for actin. And if you look with an electron microscopy, you can see thick layer of actin. So you got actin filaments, and then you've got myosin 2. And myosin 2 is going to be moving these things past each other. And as it's moving it past each other, it's going to actually cause this thing to get thicker and thicker and also become smaller. Okay, so it's becoming smaller. And eventually, you'll get a big bundle of actin here, and one cell and the other cell attach this big bundle of actin. That actin goes into something called a residual body. So basically, when the cell divides, it leaves behind something called a residual body that has that actin, the contractile actin in it. Okay, it doesn't end up in either cell. Okay, so the way that this contraction works, again, is through myosin 2, and there's, what you're looking at here is myosin 2 heavy chain. So it's actually doing it, but what you're not seeing is that right at this head area, where this is the ATPase that's moving, right at this head area, there's something called light chain, myosin 2 light chain. So you're not seeing it, but it's a regulatory subunit, okay? So the light chain determines whether the myosin is active or not, okay? And that light chain is phosphorylated by MPF, which is CDK1 cyclin B, the mitotic cyclin. Okay? So here's myosin light chain. It's the regulatory subunit. And it has an activating phosphorylation site, but it also has an inhibitory site that keeps it inactive. And that's by MPF, mitotic promoting factor, maturation promoting factor, CDK1, cyclin B. It's all the same thing. It's the CDK complex that's driving mitosis. It phosphorylates the light chain, keeps it from working. So as soon as cyclin B is degraded, okay, this site is no longer being phosphorylated, and it gets dephosphorylated because of lots of phosphatases in the cell, and now this thing becomes active. Okay, so that's why cytokinesis only occurs after cyclin B is degraded. Okay, that makes it so that it only occurs after that, because before that, the whole that myosin 2 complex is inactive by MPF phosphorylation. Okay, so now we're going to talk about this other checkpoint, which is the G2 to M phase transition. And what happens is if a cell has gone through S phase but hasn't finished replicating, it has unreplicated DNA, the cell doesn't want to go in mitosis. The cell wants to finish its replication. So this is called the unreplicated checkpoint, or if there's damaged DNA, it also wants to stop, okay? Unreplicated or damaged DNA checkpoint prevents the cell from going into mitosis. And I already told you how the cell gets into mitosis, right? The cell gets into mitosis because CDC25 phosphatase removes the inhibitory phosphorylation on CDK1 or CDC2, you can call it either one. When it removes that inhibitory phosphorylation, you got that positive feedback loop that it activates CDC25 even more, inhibits WEE1, the kinase that was putting that on, and now you've activated them all, you're in mitosis, okay? So if this is how you normally get into mitosis, well, then this is going to be how you're going to regulate that checkpoint, okay? So what happens is that when you have replication forks, you got so this is not part of the class, but there's kinases get activated there, ATM and ATR. They're going to phosphorylate CHEK1, which is a kinase. So their kinases, they phosphorylate CHEK1, another kinase. Now CHEK1 is active. So this is part of the class. CHEK1 becomes active under DNA damage by the action of another kinase, and it's going to phosphorylate CDC25, which is the phosphatase that removes the inhibitory phosphorylation, and it's going to inactivate it. And if you don't remove that inhibitory phosphorylation, you're not going to get active CDK1 cyclin B, which is MPF, and you're not going to get into mitosis. Okay? So as long as there's DNA damage, any replication fork, which you'll get from DNA damage or you'll get from unreplicated chromosomes, then CHEK1 will make sure that CDC25 does not become active, and you can't get into mitosis. All right. So now we're gonna switch to cancer. And I've bundled both of the cancer lectures together. So do we go to 4:45? 4:45 is the class? Okay. Oh, hold on a second. Okay, so all right, so we're talking about cancer, and so cancer, as you know, is the cells in somebody's body over-replicating or replicating inappropriately when they shouldn't be, and that's what actually cancer is. So it's your own cells, if you get cancer, it's your own cells over-replicating. I'll knock on wood for all of you all. So, okay, so here we're looking at the diameter of a tumor. These are talking about solid tumors here. Leukemia, of course, is just like cancers that are in your blood, like white blood cells, and they're floating around, so there is no solid tumor. But here we're talking about solid tumors, and then this is how many population doublings you need if everybody were, in fact, capable of further population doublings. But as we'll see at the end of the lecture, that's not really the case for most solid tumors. So there'll be a lot more population doublings than this, okay? At about 10 to the 8 cells, which is 100 million, you can first see it on x-ray, but you can't feel it. And then at about 1,000 million, 10 to the 9 cells, you can you'll be able to feel the tumor. And then if you get 1,000 times more, then the patient almost always dies because there's just too many cells. And, of course, as we'll talk about later, death doesn't come necessarily from a tumor growing in one location because, you can just cut it out and it's only affecting that part of the body. Generally, unless it's a critical organ like a lung or something, right? Generally, it's going to be proliferating in different places. We'll talk about metastasis. And then the proliferation basically shuts down that organ. The organ can't work anymore, and you die from the organ failure. That organ's not doing its job anymore. Okay, So cancers, as I said, is caused by unregulated cell proliferation, and this doesn't mean that they're dividing faster than normal. It just means that they just dividing when they shouldn't be dividing. A lot of cancers don't divide faster; they just keep dividing. So ultimately, it's going to be caused by mutations. And so, agents that cause mutations are called mutagens. And so generally, mutagens are carcinogens, okay? And that's just because while any one mutation may not cause cancer, because cancer is caused by mutations, if you're just randomly mutating things, well, at some point, that's going to be cancer promoting because you might hit genes that could potentially cause cancer when they're mutated. Okay, so MDs classify cancer based on their tissue of origin. So carcinomas, which are about 90% of human cancers, come from epithelial cells. Sarcoma is connective tissue or muscle. Leukemia is blood cells or nervous tissue, so sort of weird. And lymphoma is lymphoid cells. Okay, so these are just general classifications that depend on what type of tissue the cancer arose from. Okay, if we look at different cancers of people around the globe, we can see that there's big differences in frequency. So this purple super high rate, and this blue is low, and then this no data here, and then this is the thing. Okay, so if you look at liver cancer, you can see that a huge percentage of Mongolians have it, and then China has some. But if you get into the U.S., or South America, they're pretty low. Certain countries are super low. And then like esophageal cancer, you can see that like Chinese are relatively high. And then breast cancer, you see it pretty much tracks wherever Caucasian women are. So you may not know it, but Argentina is almost all Caucasian. I think they pushed out almost everybody else. So it's different from the rest of South America. And then you got Australia here, British colony, Europe, Canada, U.S., another British colony. So that's interesting, right? Like, so you think, oh, well, some of this stuff might be genetic, and clearly a lot of it is genetic, right? But there's also a lot of environmental factors. So here we're looking at whether a cancer goes up in frequency or goes down in frequency. So these blue arrows are going to be lower cancer rates, and red arrows are going to be higher cancer rates. Okay, so so if we go from Japan to Hawaii, we see that the rate of stomach cancer goes down. Okay, and then nasopharyngeal cancer is super high in China, and if Chinese migrate to America, and now they're Chinese Americans, or they have kids here or whatever, the rate goes way down. Okay, so what this is telling you is that it's not the genetics per se, right? It's not that Chinese people are always going to have high nasopharyngeal cancer. It's that there's something about being in China that'll give you nasopharyngeal cancer, whereas if you were growing up in North America, it'd be lower. Okay, and then you can see there's something about if you go from Eastern Europe to North America, where breast cancer increases. So we're talking about two Caucasian populations, but there's something about being in North America where you're more likely to get breast cancer than you were in Eastern Europe. Okay, that's not to say that there isn't genetic components, because clearly there are, right? Genetic components, and then this is just saying that there's a lot of non-genetic components also. All right, so there's these mice studies that have talked about the kind of things that can cause cancer, and they're going to break it down into two broad categories. So we're not getting into the molecular details. We're just talking about like chemical agents that can cause cancer. So there's tumor initiators and tumor promoters. So these can be compounds, and what they find is, so the way they do these studies is they take mice, they shave off their fur, and they add the chemical. And then like maybe it's a tumor initiator. And then they wait a day or so, and then to that area where they took off the fur, and you add the tumor promoter. And then the next day, they add more tumor promoter. And the next day, they add more tumor promoter. And like that. And then they just ask, well, after I do these kind of things, do I get cancer or do I not get cancer? Okay? And they're just talking about cancer in that area of the skin, because that's where they're treating it, right? So, if you put a tumor initiator on, and then you put a lot of tumor promoters after it, then almost invariably that mice gets cancer. If you do an initiator and you wait weeks and then you do this promoter in succession like this, you will get cancer. The mice will get cancer. And if they do this tumor promoter first, and then they only do the initiator later, well, they don't get cancer. And then if they do the initiator, but then they just spread out the promoter, mice generally don't get cancer either. But if you do the initiator enough, then they do get cancer. Okay, so right now you're saying, well, what's the difference between an initiator and a promoter? So the initiator is going to make mutations. So the initiator is a mutagen. It's going to be mutating the DNA. And a tumor promoter is going to make the cells divide. Okay, so just making a cell divide by itself doesn't cause cancer. That's just normal cells dividing. So you have to have the initiator first, okay? And then you have to make them divide. Like here, you're not making them divide enough. But, here you are. So you can be a lot of time in between, but then you got to make them divide a lot, and you get cancer. The same thing here. Whether there's a lot of time, not a lot of time, or a lot of time in between, it's still okay. But if you make them divide first and then do the mutation, no cancer. And here you're evidently not making them divide enough, there's no cancer. But if you mutate them enough, whether or not you make the cells divide, then you will get cancer in this mouse model. Okay, so the question is why? So these tumor promoters that are making the cells divide, why do you need them? Okay, why can't you just mutate the DNA? So there's at least two mechanisms at play. Okay, so the tumor initiator is going to introduce mutations in the genome. So what if you have a gene that is normally involved in making that cell divide? Okay, it's causing the cell to get divided. But the cell's not dividing. It's a resting cell. Well, if that's a resting cell, that gene that normally makes the cell divide is not being expressed. And so you actually have to make the cell divide before you even make the mRNA from that gene, and only then, now it makes it over divide, okay? So one is the expression, okay? And then there's a second thing that's involved, and that is that it's very difficult to make a cell that is in G0 divide. So you remember we were talking about the different stages of cell cycle. So G0 is this resting stage. Well, it's very difficult to make these cells divide. In fact, there's hardly any single mutation that will make a cell in G0 go back in the cell cycle, okay? The only thing that seems to work like all the time is there's this thing called SV40, it's a virus, T antigen. SV40 T antigen, you can put it in any cell and it'll become cancerous, and if it was quiescent in G0, it'll make it go back in the cell cycle, okay? So we'll talk about this protein later, but it's sort of unique. Most of them do not do this, okay? Most of them, it's very hard. So another way to think about it, what I would think about it is this. If the cell is dividing, it's relatively easy to keep that cell dividing, okay? Little tweaks will make the cell continuously divide and not go into G0, okay? But once a cell is not dividing, it's very hard to get it back in the cycle. So clearly, that's why it's going to be way easier when cells are dividing to keep them dividing, which would be one of the reasons why you need the tumor promoter, to get them going through the cycle. And you need very little mutations to make them go. But as we'll see, in cancers, things are pretty complicated, and there's usually, in solid tumors, usually multiple things are mutated. Okay, so this is an example of one of these tumor promoter. So this so you've seen this in signaling pathway, I hope, in this class. There's diacylglycerol, which is required to activate protein kinase C, and it's coming from this PIP2 getting cut, and then it activates protein kinase C. So it turns out that if you add something that looks like diacylglycerol, called 4-bil esters, that'll also make the cell divide too much, okay? So some of these things just look like other signaling components, and they're going to make the cell divide. Like if you were to give it diacylglycerol all the time, it would divide too much too. This is just like an artificial diacylglycerol. Okay. So now we're going to talk about clonal expansion. So generally, tumors are clonal. So what that means is that if you see a tumor, it arose from a single cell at one point. So, and you get this clonal expansion by going through these different stages of cancer formation. Okay, so the first stage is where the cells are dividing, but they're sort of acting normally. All right, and in this stage, we're going to give it a name. Okay, and that name is hyperplasia. So plasia is sort of like, the kind of term they use for cancer. Hyperplasia just means over-proliferating. But they're not actually cancerous, not cancerous. It's not cancer. And then another term, so these are the same thing, okay? So this is not cancer, but it's over-proliferating. Okay, so what does that mean? Okay, let's say we had a cuboidal epithelium. Okay, so an epithelium is like usually a layer of cells, and cuboidal means that they have this kind of a structure where they look like little cubes lined up. Okay, what happens if you had hyperplasia? So hyperplasia, you're going to have more of these cells. Okay. You might have them squeezed in more. You might have them pop out a little bit, okay? But it doesn't change the fact that these cells are still cuboidal epithelial. This is an example, because it's easy to draw these cuboidal epithelia, these little squares. Anyway, hyperplasia would be that these cells are basically acting the same. They're still cuboidal epithelia. There's just more of them, okay? So that's hyperplasia. And so all the constraints on that cell are still going to be there, okay? It's still going to act like cuboidal epithelia. If it's doing something, it'll just do the same thing, okay? There's just more of them. So generally, it's not necessarily a problem, hyperplasia, dysplasia, but it is the beginning part of this what's called neoplastic progression or cancer progression. Okay, so that's what we have here. These cells are still normal. These ones aren't cubes, but you can see it looks about the same color. Well, it's close. Anyway, now you've got more of these cells dividing in hyperplasia or dysplasia, and then you can have a bigger population for other mutations, and these other mutations may make it divide faster. Okay, and then in this growing population, you might have other mutations that allow it to grow faster, even faster, right? And so because you think about it, there's always a positive selection for these things. Why are there more of the red cell? Well, that's because they were able to grow faster than the orange cell. Why is there more orange cell than these light green cells? Why? Because they're able to divide faster. So anything that can divide faster or divide further or in a different place, it's going to just outcompete the other cells. Okay. So they have this clonal expansion, but as you're doing it, the thing is changing. The cells are gaining different mutations or epigenetic changes that are allowing them to outcompete the other cells before them. Okay, so here we're seeing, like I said, hyperplasia, dysplasia, cells overproliferate, they're still acting normal. But neoplasia, the cells are overproliferating, but they no longer act normal. All right, what that means is that whatever they were before, they're no longer like that anymore, okay? So these cuboidal epithelial cells, no longer cuboidal epithelial cells. And we'll talk about how they're gaining these different characteristics in a little bit. So when you're talking about neoplastic progression, we're talking about, like here, you see hyperplasia, but these cuboidal epithelial cells are still cuboidal epithelial cells. Now suddenly they get other mutations. Now they're starting to proliferate outward, okay? And now, even more mutations, now they're invading other tissues. And you can see that what this yellow is a excellular matrix. So excellular matrix is like, it can be thought of like this sheet that's usually separating tissue compartments, okay? It's a way for the cell to like, okay, we're gonna have this one organ in here, and we're gonna separate it from the rest of the body by excellular matrix. And so cells generally stay on one side, right? I mean, well, they have to, because that's how the body's organization set up, you know? You got this thing, this keeps the cell, this keeps the organ in place, everybody else is not touching it, okay? But some of these cancer cells at the later stages, when they're called metastatic, they can, malignant. So malignant means that they're now invading other tissues. And as part of that, they often can break through the excellular matrix. And now, and so, normally these cells could never do that, right? Well, they would never be able to do that. It's not part of their job description, right? Being cuboidal epithelia. They're supposed to stay on one side, act a certain way. But these cells have now acquired other characteristics, allow them to invade other areas. So that's the neoplasia. So this is malignant if they invade other sections, and they're also called neoplasia or cancer, whereas this is really just like a pre-cancer thing, this hyperplasia or dysplasia. Okay, so this process is called neoplastic progression. Here you see this epithelial layer, and it starts over proliferating and dysplasia, still acting normally to some extent. And then a carcinoma in situ is where it really starts invading other sections. And in situ just means in place. And then a malignant carcinoma is where it invades other areas. So I guess the carcinoma in situ is it's no longer acting like the normal cells. And then when it's malignant, it's invading other areas. And this just shows you the normal situation. You got this excellular matrix you're not being able to see here, which is separating this tissue from this one. They're totally separated by excellular matrix. But now, once you have a malignant carcinoma, it's now invading across that boundary. And clearly, if you're at a carcinoma in situ, or even if you're malignant, but you haven't really gone anywhere, I mean, it's relatively easy to treat that. You just cut it out. You would just cut out the whole thing. Not a problem, right? Unless it's in your brain. And then they have trouble, cutting out big sections of your brain. But for most things, if it stays in one place, that you just treat it with surgery. And you just, as long as it hasn't spread somewhere else. Okay? So that spreading to other parts of the body is called metastasis. And so, you can have it's a malignant tumor because it's invading other areas, right? It's gone through the basal. So this basal lamina is just another term for excellular matrix. Okay, excellular matrix, basal lamina, same thing. So it's invading here, it's malignant, but it hasn't metastasized yet. It hasn't moved through the body either in a blood or in a lymph node. So lymph is like a way, is a system that puts lymph fluid throughout the body, allows your immune systems to have like a expressway they can travel along and other things. So it's also another system that will connect different parts of the body. So you can get metastasis through the bloodstream or through the lymph system. At any rate, here we're looking at the blood cell. So one, it's got to get the capability to enter the bloodstream. So it's got, these are endothelial cells. So on the side so blood vessels, are not just like some kind of like basal lamina or something. It's actually cells, okay, endothelial cells. And they're connected to each other. And so these cancers have to be able to slip past those endothelial cells to get into the blood supply. Then they have to attach to the side of the blood vessel, which again is endothelial cells. And they have to be able to get out. So they have to be able to get in, and then they have to get out. And then they have to be able to start, proliferating in some new location. Okay? So this is an awful, and also, remember, they didn't even have the capability to get through the basal lamina before. So this is an awful lot of new capabilities that the cancer cells have to pick up, okay? They got to be able to cross the basal lamina, excellular matrix. They got to get into the blood supply. they got to attach, and then they got to get out, and then they have to be able to proliferate in the new location they're not used to. So these are some of these properties that I told you about. You got to dissociate from neighbors. That's not really true. Here you're seeing individual cells going out, but it's not really true. If you take, so I don't know why I have a slide on that, but at any rate, it's old evidently. You take a mouse and you shave off its fur, and you put cancer cells below the surface that have green fluorescent protein, and you put a camera right on that, okay? Then they're able to see the metastasis occurring. And generally, when it occurs with sheets of cells. So it's not like individual cells moving. No, the cells stay attached, and they move off as large sheets of cells going off, streaming off in different directions. So it's not really true that they have to dissociate from the neighbors. They can, but they can also just learn how to move as a sheet. Okay, so, so one thing is they have to start moving. A lot of these cells don't move, right? And in fact, depending on how many of these capabilities they have, depends on how easy it is to get metastasis for that thing. So one thing's not even listed here is they got to be able to move. So that's one of the reasons why, skin cancer, melanocytes, what's it called when you, melanoma. So one of the reasons melanoma is so dangerous is because these cells, these melanocytes, they always move, okay? That's part of their job description, to move in the cell. They move, they'll make a freckle, they'll move this way, they'll move that way, okay? They're constantly moving in the skin. So if they become cancerous, well, they already can move. They don't have to get that capability because they're already a motile cell. So depending on how quickly these things can be picked up, depending on how quickly they can metastasize, and if a type of tissue already has some of these capabilities, clearly they're gonna have a head start on doing metastasis, like the melanoma does. Okay, they got to be able to cross the basal lamina, which is extracellular matrix. They got to cross that endothelial lining, and then they got to get out as well and start in a new location. So you can see that when you're talking about these capabilities, what you're really talking about is picking up molecular, the ability to do things at a molecular level. So like the cell's got to be able to be able to cross this barrier. First, it's got to be able to attach to it. So if they weren't expressing the laminin receptor, which is laminin is a part of the extracellular matrix, then the laminin receptor allows it to bind like to collagen. So if it doesn't have that laminin receptor, it can't actually attach. If it can't attach, can't do anything. Then it's got to be able to express things to degrade it. This thing is like a sheet of plastic, It's like tough plastic. The cell can go in, it's just going to bend out and stuff like that. To actually get through it, the cells are not going to burst through it, okay? It's just too tough. They're going to have to degrade it. And you're going to degrade it by, so these are actually proteins, like collagen's a protein. So you express collagenases, which are proteins that degrade collagen, and that will allow them to get through it. So they have to pick up the capability to attach, and then they got to pick up the capability to make collagenase, to degrade through it. And then, like I said, they got to be motile. And as we said, having any of these things gives them a head start on being a metastatic cancer, because then it's more easy for them to move around. And then this is another term which we haven't gone over yet. So when you hear a benign tumor, that means that the thing is no longer acting normal, okay? It's not acting normal, but it hasn't invaded any other tissues, or at least it stayed in one location. So that's benign. And also that means that unless it's in your brain or some other really critical organ, you can generally just cut it out with surgery, and there you go, you solved the cancer. All the cells that were dividing too much are now gone. Okay? Metastatic, the problem, of course, is they could make many places in the body, and then it's suddenly hard to get rid of all of them, and you can't even see where they all are because they're spread out. Okay, so we will get into exactly how these cells are no longer normal in a little bit, in a few slides. Okay, so what's happening, so, but basically one of the concepts is that you need to have some genetic instability. What does that mean, genetic instability? That means that, like normally you have one copy of every gene and everything is acting normally. Genetic instability is where things are no longer normal. You know, parts might be overexpressed, underexpressed, change expression, okay? This kind of genetic instability is going to allow them to pick up these kind of attributes that they normally don't have. So you're basically taking a cell that's acting normal. That cell acts normal. That's not a problem. You know, it's going to be a cuboidal epithelia. It's acting normal. And then you're taking it, and now it's getting new capabilities. Well, genetic instability is going to contribute to picking up these new capabilities. And if you have too much genetic instability, well, all the cells are going to die because they're going to start losing or gaining, losing genes that they need or gaining toxic things or whatever, right? It's just too much genetic change. And cells that are being made are dying, you know, and you're not going to generate a cancer because the cells are just going to die out because there's just too much change. Like if you took away somebody's entire chromosome 6, I mean, that's probably toxic to the cell, right? I mean, I don't know that for a fact, I'm just guessing, but I'm assuming it is. Any one chromosome, you know, if you get rid of all copies of it, it's gonna, you know, they need it for metabolism and all these other things. But anyway, if you haven't, as far as the cancer's perspective, there could be an optimal genetic instability that allows the cancer to pick up new capabilities without like going crazy and just losing all these essential genes. And then you know, there's still some genetic instability that allows it to get new capabilities, and new genetic, and then allows it to get through these barriers. And these barriers don't have to be a physical barrier like a basal lamina. It could be just like a type of barrier like, oh, the ability to move, or the ability to go through endothelial cells and to get in bloodstream, whatever the barrier is. Or, you know, we're in the middle of a cancer, a tumor, and it's very low oxygen. So how to deal with low oxygen, right? Or there's no blood cells because there's low oxygen. How to get new blood vessels coming in. All of these are like barriers that are going to limit the cancer proliferation. But, you know, one cancer cell might get the answer by having the right genetic combination. And then it'll outgrow. And again, we're talking about if the cancer cell can outgrow, then it will. It'll leave all the other ones behind, and it'll just keep dividing. Okay? So it's part of that cancer progression. All right, so what we're talking about here, cancer cells are usually aneuploid, but we're only talking about solid tumors. We're not talking about leukemia, okay, not leukemia. Leukemia doesn't have so many of these barriers, okay? Leukemia, if you just have too many of the cells, you're gonna have leukemia. They're just floating around in a blood vessel, but if you have too many of them, it's gonna cause problems, okay? But they don't have the same problem with the barriers. They don't have to worry about extracellular matrix. They don't have to worry about motility. They don't have to worry about anything. They're just floating around your blood vessel. But anyway, so we're talking about solid tumors, and solid tumors are usually aneuploid. So here, this is a karyotype analysis. What do? You take the cancer cells, and you drop them, the mitotic ones. You can like add nicotazole to make them all mitotic, and they're arrested in mitosis. You can drop them onto slides. You fix the slides, you stain it, and then you line them up. And people always line up from chromosome 1, which is the largest chromosome, to like 22, and then you got the sex chromosomes. They're the smallest ones. And you line them up in order, and then this is like gene sustain, where there's particular banding patterns. And what you see is that there's whole things missing. You're like, oh, well, what happened to those chromosomes? Like, evidently chromosome 4 and 5 are missing, right? So you're like, that's messed up. You know, and then there's more of certain chromosomes. But it's not like they really lost those chromosomes, because like I told you, if you lose the entire chromosome, those cells are probably in a bad way, right? They're probably losing essential proteins. And you only have but so much resolution with gene sustaining. It's just looking at chromosome patterns. And these are patterns of heterochromatin and euchromatin, which also can change during, you know, in different cells. Okay, so here it's called painting the chromosomes. All right, so what you would do is you would have a certain color. You would get chromosome 1, and you would make probes from chromosome 1, and you'd make probes from chromosome 2, And we're talking about chromosome-specific probes, not repetitive DNA. You have chromosome 2 probes, so a piece of DNA. Chromosome 3, chromosome 4, chromosome 5. And then you come in and you add particular dyes to it. And in fact, we don't have that many dyes. There are not that many dyes. So you might have like 4 or 5 dyes. So you would add different ratios of the dyes. So you would have, I'll add the green and the red in a certain ratio, and that'll be this chromosome. Or I'll add, yellow with green, and that'll be this other chromosome. So you're mixing them together because you don't have 20 dyes that are different colors. We just don't have it. You might have far red and different things like that. Anyway, and you do this karyotype thing where you're spreading out the mitotic chromosomes, and now you come in, you denature the DNA, okay? So it's double-stranded. Now you make it single-stranded, and then now that it's single-stranded, you can hybridize these probes to it. Okay, so you hybridize the probes, and now suddenly you can tell where everything came from. Like I don't know, but maybe this magenta was chromosome 1. And what you see is, there's one here, there's here, but there's also one here. So like suddenly you can count up and see where it's happening. And here you have this like light green, but you have light green here and light green here. And then like this type of green, olive green, well, it's here, here, and here. So suddenly what you see is that while it looked like whole chromosomes were missing, what happened instead is that chromosomes are starting to fuse with each other, okay? And so the normal patterns disappear, and you're having more, like this, look at this nice, beautiful color of blue. Well, there's a ton of it, right? This part of the blue, this chromosome is overrepresented. Other chromosomes may be underrepresented, okay? So this is what's causing the genetic instability. And in the next lecture or the lecture after that, we'll explain exactly how this starts. There's a particular mechanism that will cause this. But for now, all right, I have to stop now. But there's a super interesting thing about this, but I'll talk about it next time. Anyway, Snehal will be in charge of giving out the exams.
What were you going to talk about?
I was going to talk about the. I'm going to stop this now.